Hi, this is Miss Litton, and this is 7th period review with a bunch of AP Bio students. Say hi. Hi. Hope you had a lovely day today. So we're going to try to focus just on Unit 1 today. So a view of life, that was our big overview where we kind of understood like all the big ticket items and the big ideas we were going to discuss. Basic chemistry. If you haven't already revisited basic chemistry, I want you in your mind to think, where should you start with basic chemistry? An atom. Excellent. Okay. And then that seemed pretty good. And if we had an atom, we would want to know about the structure of an atom, right? And then we'd want to know why one atom might bond with another atom and how they would go about doing that. And why one molecule reacts with another molecule, because if we understand the chemistry, we can understand the biology, the name of this class. Okay? So that was basic chemistry, and then in chapter 3, we focused, there are inorganic and there are organic molecules. What are the 3 important inorganic molecules? Oxygen, carbon dioxide, and water. And then there are 4 important organic molecules. Why would we even want to understand those 4 important organic molecules? Because those 4 important organic molecules are going to build what for us? Cells. AP. Yeah. Those are going to build cells for us. And we know that all living things are made out of cells, and that they are the structural and functional units of all life. Yeah, and cells only come from pre-existing cells, right? Through binary fission or mitosis or meiosis, right? So we just need to make sure we remember how we put all of this together and how it fits into what we know, okay? So that's what we were doing here. So chapter 1, very first thing, okay, the very first thing that we talked about in chapter 1 was the characteristics of life. Let's super fast go through those. Living things are organized, made of cells. Doesn't that give you a happy memory? Okay, here we go, ready? Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. So many ways, right? Positive feedback, negative feedback. They maintain homeostasis. They respond to stimuli, maybe with a receptor. They respond to stimuli and reproduce sexually or asexually. They grow through mitosis and they adapt, evolution, and die. No longer maintaining homeostasis, no longer able to fight the good fight of entropy. All right, now, we then talked about these 4 big ideas, and they keep recurring throughout. If you look at your master hyperdocs, I have in there, right, the different connections with the big ideas. You might want to look at that. So the process of evolution drives the diversity, how it changes, adapts, diversity, and the, what does it say? It's right there. Unity of life. Unity of life. That makes me think of Luca. What are the 3 big pieces of biochemical evidence that we talked about supporting Lucanus? DNA, RNA, and ribosomes. Okay, the third one was metabolic pathways and signal transduction. What was the second one? The central dogma, right? And that's how DNA goes to RNA and builds proteins. Where are the proteins built? Okay, just checking. Okay. Big idea too. Biological systems utilize energy and molecular building. Oh, what are the molecular building blocks? Carbohydrates. But what are those built out of? What's the building block? Sugars. Okay, give me another one. Lipids, 2 categories there. Fats and what are the building blocks of fats? Fatty acids, which could be saturated or unsaturated, and they're attached to what? Glycerol, right? And what's the other lipid? Made out of 4 rings of. See, this is chapter 3. You already know it. Okay. Carbohydrates, lipids. Give me another one. Proteins. Proteins are built out of amino acids. Amino acids. You remember their structure? What's up? Not phosphate. Hydrogen. Hydrogen. Okay. What's over here? And what is that? NH3. What's over here? Carboxyl group, C double bond OOH. What's below? Here you go. R group. What is it? R group. How many different R groups do we have? 20. Hence 20 different amino acids. Put a pin in that. We'll come back to it. That was our third important organ. And what was our fourth one? Nucleic acids, which are built out of nucleotides, which are built out of a base, sugar, and phosphate group. Okay. So biological systems utilize energy. How do they get that energy? Where does all the energy come from? The sun, ultimately. Who captures that energy? Plants. What are they called? Autotrophs, right? So plants capture that energy, and then that gets passed to what? What do we call it? Heterotrophs. What's another name for it? Consumers, right? Consumers. All right. And so then we can use it. How can we use it? Well, plants capture it through the process of what? Photosynthesis. So plants do that where? In their chloroplasts. Okay, good. Using what pigment? What was the first reaction called? Light dependent. In fact, the one that's used today is non-cyclic photophosphorylation, right? But non-cyclic photophosphorylation didn't evolve until about how long ago? Oxygen revolution. When was that? 2.3 billion years ago. You're like, okay. Okay. What was the second reaction called after the light reaction? The dark reaction, or the Calvin cycle, or the light-independent reaction. Exactly. Okay. Then plants make those sugars, right? And then plants or animals can use those sugars through what process? Because we have to utilize the energy and the molecular building blocks. What's the first step? Who does it? All cells. All cells. That makes me think of, my name is right? Okay? Yeah. Last universal common answer because it's done by all cells. What's the second reaction? Prep. What's the next reaction? Citric acid cycle. Give me another name. What's the next reaction? ETC, where you make a bunch of ATP. Who catches electrons at the bottom? Forming water. What if you don't have any oxygen? Aerobic respiration. Not aerobic, but anaerobic, which is called. How many choices do we have? 2. But either way, we. Exactly. So that's number 2. Okay? Big idea number 2. Biological systems utilize energy and molecular building blocks to grow, reproduce, and maintain homeostasis. So many different ways to maintain homeostasis. We spent a lot of time on that. Living systems retrieve, okay, transmit, and respond to information essential for life processes. What are some things you could use to receive that information, deal with that information, and respond? Your nervous system, right? What else? Hormones will do that. Signal transduction, right? Any kind of chemicals. How about if you're not multicellular? Form sensing. Yeah, good. All right, those are all examples. What did you say? Yeah, worms. Yeah, worms. Worms do it. Biological systems interact, and these interactions possess complex properties. Let's talk about the organization of these complex properties. It starts with atoms, molecules, organelles, cells. What's this? Life starts. Tissues work together to form. Which work together to form a? Which work together to form a? If you have several organisms, you have a population, community, ecosystem, biosphere. Okay? We're going to talk about population, community, ecosystem, biospheres in one of our last units that we do this school year. Okay? So those are your 4 big ideas. You already have a little taste of all 4 of those big ideas and would be able to write on them. All right. Next, we totally know this. Look at this picture. It should all make sense to you now, yes? Life shares the same molecules and reactions, and it points to a common ancestor. What's going on up here? What is that? Those are amino acids joined together to form a... What's that bond between 2 amino acids? Peptide bond. Good. What's the reaction to form a peptide bond? Dehydration synthesis. Another name for that? Condensation synthesis. Will water be a reactant or a product? Dehydration synthesis. One happy sugar, another happy sugar. They're living fine together. If I want to join them together, I have to take something away from each one of them. From one, I will take away what? Hydrogen. From another, I'll take away oxygen and hydrogen, forming a water molecule as a product, right? And a disaccharide as those 2 sugars are joined together. Yes? So is water a reactant or a product of dehydration synthesis? Product. If I go backwards, if I want to break those sugars apart, what's that called? If I want to break a phosphate off of ATP, it's hydrolysis. I have to input that water to break it apart, but I release energy, yes? Okay, all right. So there I can see, here's a nucleotide. What's this yellow thing over here? Here, I'll make it bigger. A phosphate, good. Okay, and here is a sugar, and here is a... Could you tell me if that nitrogenous base is a purine or a pyrimidine? Pyrimidine. How do you know that nitrogenous base is a pyrimidine? It only has 1 ring. Who are your pyrimidines? Adenine and... No. Okay, give me this. Who are your purines? Adenine and guanine. So who does adenine like to bind with? Thymine. Who does guanine like to bind with? Cytosine. Those are your pyrimidines. And what's your 3rd pyrimidine? Uracil. Do we have uracil in us? Yes. In all of our what? RNA. Did you forget you have RNA? You use RNA to build proteins. Okay. Now, how many hydrogen bonds do I see between guanine and cytosine? Between adenine and thymine. And those are building a what? Chromosome. These would be called what? Sister chromatids. When do I see sister chromatids for the first time? Prophase. When do they line up in the middle? Are they lining up with their homologous pair? Only in meiosis. What stage of meiosis? Metaphase 1. And they can do it independently of each other. That's called... Okay, good. All right, just checking. We will move on to here. Oh, there's sugar. What's the simplified version of all the molecular formula of all carbohydrates? CH2, because we're going to remember that carbohydrate. And why did they show a carbohydrate here? Oh, Lord in heaven, in my heart. What is that? It's a chloroplast. What do chloroplasts do? Listen, my friends, the reason I'm reviewing this way is to stitch it all together for you. You need to think of it differently, not as individual factoids, but you got to see how they all connect, right? Because when you take your AP exam, they are not not going to think of it like this. They're going to think about all the information you're expected to know. So start to look at that and go, of course. And that must be making what? Starch. But we don't store carbohydrates as starch. What do we store it as? Glycogen. Where do we put it? Liver. What would be one way to release that glucose from our liver? What? Insulin would increase glucagon, right? What's another thing? Epinephrine, because what's another name for epinephrine? Adrenaline, because I might have what? Fight or flight. All right, just checking. Yay, fat. Okay, what does this represent? Hey, I'm here. Talk to your friends later. What is this? What is this? Glycerol. What are these? Fatty acid chains. Fatty acid chains. Good. It looks very straight. What kind of fatty acid chain is this? Saturated. Good. All right. All right. Good. Now, before I go any farther, tell your bio buddy, little dipstick, checking the oil. What right now do you know you already need to review? Go. All right. Enough of that. We already know this, right? We know. Okay, talk to your friends later. Last universal common ancestor, we feel comfortable. Look at this. Bacteria, archaea, eukarya. Could you give me little one-word descriptors about each one of these things on here? Yes, you could. Right? Yes, you could. You would give me, you would say, oh, archaea and bacteria are both, but archaea live in street, whereas bacteria live everywhere. And eukaryotic cells, they all have membrane enclosed organelles like the nucleus, right? And if I looked at these, I'd be like, animal. How could I describe all the animals? They are all multicellular. They are, what kind of heterotroph? Ingestive. Did I not teach you that? Oh, I am now. Ingestive heterotrophs. Ingestive. Ingestion, digestion, absorption, egestion. All the shissions. Okay. Fungi are absorptive heterotrophs. Did we not learn that? No. Are you sure? Yeah. Okay, I'll look at my notes before I write your test. Okay, plants. What are plants? Autotrophs. Good. Algae, that's a little separation out there, and that's because that's old school, those kingdoms. There could be one called Chromista. I'm not testing you on that, so don't stress out about that. We haven't covered that yet. All right, last universal common ancestor. We spent some time talking about that last class. Let's move on. Oh, that's funny. Okay, so natural selection is the mechanism for what? Evolution. Have we had a unit on evolution yet? No. But you do know that evolution is change over time. And the best adaptations, those are the ones, those individuals are going to survive and reproduce. Right? Okay, good. All right. Big idea too, cellular processes, energy, and communication. Have we talked about both those things? Yes. All things energy, all things communication. You want to remember that when you look at, we're going to hit this at the end of the school year, but just the basic part of this is energy flows, remember this, and nutrients what? Cycle. Energy flows and nutrient cycle. Now you know something about energy. Every time, what's the first law of thermodynamics? It can only, and every time it changes forms, you lose some as heat. Okay, so you know that. Good. Homeostasis we have discussed. Big idea 3, genetics, information transfer. We are working on that right now. Okay, but the big idea there is that your information is stored in your DNA. You take a copy of it, and that is what? mRNA. And then you use that mRNA in a workbench. What's the workbench? To build a protein. There you go. All right, and we'll talk about behavior as well. That's a form of responding to stimuli. Okay, big idea 4, we have already done this. Okay, we already went through that, so we don't have a problem with that. Emergent properties at each level, we've gone over that. Atom, molecule, organelle, cell, tissue, organ, right? You gain new properties as you move up. Let's see. Okay, then we talk about the science practices. When you look at these science practices, I would just kind of review them and think about it. Most of you think about this all the time. Anyway, when I ask you about it on your test, you're like, no, I don't think about it. Okay, but if we look at science practice 1, the student can use representation. What's the key word there? Models. We look at models and diagrams all the time. Number 2, the student can use math. Give me one right now that I can use to analyze data. Thank you. Okay, science practice number 3, scientific questioning to guide investigations. What do we have at the beginning of all our lab investigations? A driving question. There we go. Science practice 4, student can plan and implement data collection. When we collect data, what is the kind of data we could collect? Quantitative and qualitative, right? Quantitative is an actual, something I can measure. Good. The student can perform data analysis. You might be using math to do that, right? Then we talk about claim, what? Evidence, reason, working with explanations. Science practice 7, the student is able to connect and relate knowledge across various scales, concepts, representations, and across domains. Okay, that you can communicate out what you have learned. You actually practice all those things all the time. Scientific method, your science notebooking, you can follow that to see the scientific method. I think you're comfortable. We have gone over this. Independent variables versus dependent variables. So which one is the one I choose? Independent. Which one is my data? Remember we talked about research versus what hypothesis? No. Research is I'm looking for a correlation. If then, right? There's a connection between the independent variable and the dependent variable. Okay? A null hypothesis, I'm saying there is, in my mind, might I think there still is a connection? Yes, but I want the data to overwhelmingly show that by being statistically significant, and that's when I use the chi-square test. Good. All right. We talked, and I'm going to hit this because we're going to hit it in our genetics unit, so I'm going to review the chi-square test with you with genetics data. Okay, so I'm going to put a pin in that because we're going to be reviewing that shortly. All right. Atoms, you already know this, right? Let's very quickly do the atom story. The atom story. Atoms are made of, you're doing it slow, protons, neutrons, and electrons. The protons and neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 1832. 8, 8, 8 is great. Octet rule, right? Now, when we talk about proton pumps, what is a proton? It's just a hydrogen what? Ion. Because hydrogen is an exception because hydrogen only consists of protons and electrons. One proton, one electron. So when you take an electron away from hydrogen, I don't know, for maybe an electron transport chain, you're left with a proton. So whenever you see something like proton pump, you're really thinking what element? Hydrogen. Okay, just checking. All right. Then we talked about happy, unhappy. Is lithium happy? No. How many protons does lithium have? 7. Really? How many protons does lithium have? 3. He has 3 protons. Hi, AP. What else does he have? He has 4 neutrons because 4 plus 3 is 7. This number will always tell you the number of protons. This number right here is the... Okay. Is lithium happy? No. Okay. Lithium's not happy because he has how many electrons? 2, 3. He has 3 electrons. How many can we put in the first energy level? 2. How many are left? 1. Where's that going to go? Is he happy? No. What will he do? He will either... I look like I'm sharing. Share. Or he will what? Give it away now. If he gives it away, what will his charge be? Negative.
Oh, yeah, that's fine.
Did we go over the Adam story already? You thought it was simple, but you're missing all these questions. That's terrifying. If I give an electron away, I have given away a negative charge, I will be more positive. Thank you. Thank you. If I am positive, I will be attracted to a negative. Thank you. Okay. Okay, Papa. Now, when we look at a periodic table, remember periods and groups? Which one's horizontal? Periods. Yeah, you know why you're remembering that, right? Periods on the horizontal, right? Groups, what do groups have all in common? Same number of, so they will behave in the same way, yes? Right? Because they will react in the same way, making the same kind of bonds. But here they're in the same what level? Energy level. Great. Okay, perfect. We talked about ionic versus covalent bonds. When you share in pairs. Sometimes you do not share pairs. If you don't share fair, what kind of bond is that called? Polar covalent. If you share fair, what is it? Which one is hydrophobic? What? Non-polar. Because non-polar, they're sharing fair, so nobody has a charge or a partial charge. So when water wants to make a what kind of bond? Hydrogen bond, there's no place for it to hook up. There's no place for it to dock, right? So it doesn't form that hydrogen bond. But a polar molecule or a charged molecule, water can interact with. Okay? All right. Moving on. Oh, hydrophilic properties of water. High heat capacity due to those hydrogen bonds. Good solvent, adhesion, cohesion. I would review that. I think we have a whole essay over that. Right? Sorry, that slide is jacked. And then we talked about pH. pH is a measure of what? Hydrogen ions, right? Measure of hydrogen ions. If you have a low pH, you actually have a high concentration of hydrogen ions, and it's called an acid. The opposite would be a base. Excellent. Okay, buffers help keep things neutral. All right, good. Now, we've actually already reviewed this, okay? Organic molecules have what? Carbon. Thank you. What's an exception to that rule? Carbon dioxide, because it's too what? Simple. Okay? So you have functional groups that you can attach onto hydrocarbon side chains, which affect their properties. And then we had a little chart. Remember a little chart? Remember the chart? You love the charts. You can take a picture of anything. This is recorded. It is for all to see. All right? So on this chart that you have right here, you can see the 4 important organic molecules. You can see the monomers, what they're each built with, right? And then you can see polymer examples and functions. Have we gone over this? Yeah, we already went over this, right? Yeah. So you should be feeling pretty comfortable about, but you should also be looking and going, what else do I know? When I look at this, when I think about carbohydrates, think about the process of making them. Think about modification of carbohydrates. It doesn't just have to be for energy, it can also be for what? Structure, and that depends on the type of what you make. Bonds that you make. What are bonds? Shared pairs of electrons. If you have a lot of shared pairs of electrons, electrons equal energy, and that's why we might burn carbohydrates for fuel, right? So there's a connection between all of that. When we look at our lipids, we look at fats and oils, phospholipids, and cell membranes. You know a lot about cell membranes now, right? Cell membranes consist of what? Proteins. What? Phospholipid bilayer. What else? Cholesterol. What's the cholesterol there for? Fluidity, right? Yeah, to prevent it from freezing or... And then what's on the outside of those? Glycolipids and glycoproteins, known together collectively as the glycocalyx, right? So when we look at things like carbohydrates and lipids, don't look at them only in this chart. Think about, where have we learned about those? Where have we used that information? Why do fats have more calories than carbohydrates? They can make more what? Okay, there are more bonds. Think about cellular respiration. Yeah, but why? Oh, I heard it. Somebody said it over there. More acetyl-CoAs. Those long fatty acid chains, right, that you have right here, those can be snipped in. Remember, glucose will make you how many acetyl-CoAs? 2. Acetyl-CoAs put 1 away. Okay? Whereas a fatty acid chain, right, if you're 18 carbons long, how many acetyl-CoAs will that give you? 18 divided by 2. 9. How many chains do you have? 3. 3 times 9, 27. Acetyl-CoAs put 26 away. Okay? Do you see how that can make you more ATP? Yeah. So don't think about these important molecules anymore in isolation of your chart. Think about them, how they relate to all the units that we are discussing. Stitch it all together. That's where you'll make your mistake, if you think in factoids instead of the big ideas. Because now you know enough that you can go beyond and start pulling everything together from all your different units. Okay? Practice doing that. Nucleic acids, we're talking about that right now. I have put out, I just wrote it last night and I posted it last night. There's a POGL. And there's a POGL on the statistics of heredity. It's due Friday morning at 8:30. That would be a good practice. You can do that with up to 3 people if you would like. I also put out there the AP progress check, but it's going to have a lot of questions about some of the genetics that we haven't discussed yet. So you might want to hold off on that. It's not due till the start of next week, and I think there's like 22 questions. And that is not so much for a grade, but that you experienced it. So you might want to wait till you can experience it better so that you can use it as more practice. Does that make sense? Okay? So 1 unit down, you need to take time at some point to reflect, where in this were you weak, right? Where were you strong? What do you need to study so that you're prepared, right, for your exam? Questions you want to ask me? Yes?
Are the College Board checks going to be like similar to the AP exam?
Yes. Though that's who wrote your exam. The hard thing about the College Board checks is this. They are asking you questions really based on a year's worth of knowledge. You don't have a year's worth of knowledge yet, so it's hard for you to stitch it all together. Okay? But those are the types of questions they are going to ask you. So you want to get familiar with them. Have you noticed that they are longer? Okay? And they're more application level when you want to do that. As we get closer to the AP exam, we're going to redo all the College Board checks, and then we'll talk through the answers and use that as part of our reviewing once you have more information in that big, beautiful mammalian brain that you have. Yes?
For the fatty acid, just for fats in general, so are you saying that one...
Hey guys, I can't hear when you're talking. Go.
Are you saying one molecule of fat will have 27 acetyl-CoAs?
Actually 28 because of the glycerol, but yes.
Okay.
Because we're going to use pyruvic acid. Okay, what else? All right, I will see you tomorrow. I'm already proud of you. Make good choices. I love you very much.